Hello everyone. Today we are going to discuss with you Plant Growth and Development Chapter Theory Part 2. In this session, we are going to discuss with NCERT section 13.3, 13.4, 13.4.3.1 to 13.4.3.5. So this is our disclaimer. So let us see the learning objective of today's session. First one, that we will going to define what is plant growth development, and that will explain about how the relationship between the growth and differentiation is established. We will also talk about the redifferentiation and dedifferentiation phenomena. Now, there are the 5 different types of plant growth regulators. So in today's session, we are going to discuss about each and every plant growth regulators, right? And we will talk about what is the bioassay of it, what is the effect of it when it is applied to the plants. Okay, now plant growth regulators act like a tool, okay? And we will talk about the discovery of it, like by what type the plant growth regulators are discovered, what is their effect, and how they are isolated. We will see the physiological effect of each major plant growth regulator. What is its role on the plant growth and development? Now, if we see the index part, then we will discuss about the development that covers the 13.3 section of the NCERT. We will also discuss about the plant growth regulators covering 13.4 section of NCERT. Then there will be the characteristic 13.4.1. Discovery of the plant growth regulators will be discussed in the section 13.4.2. And physiological effect of the plant growth regulator will be discussed in the section 13.4.3. Now, what are the plant growth regulators we are going to discuss with you today? First one is the auxin, then gibberellins, cytokinins, ethylene, abscisic acid. Now, at the end, we will summarize the lecture with a recap of today's whatever we have discussed with you. Then there will be some NCERT based questions. And there will be some keynotes. There will be some interesting question as well that you are going to solve with your own. Okay, so we will start today's session with the development. So what is the definition of the development? Let us see that. Development is the sequence of all the changes that an organism goes through during its life cycle, right? And this is the foundational concept help us understand the stage a plant undergoes from start to the finish, right? When we are discussing about the cycle, then it is showing the start stage and at the end stage. Now, it means that when the seed get germinated, right? From the seed germination to the senescence. Each stage of development is crucial for the plant survival and its reproduction, right? So, it includes both the growth and differentiation. Growth is about increase in size, while differentiation involves the cells becoming specialized for specific functions. Now, there will be the development, okay? Now, if we talk about the development means development is equal to growth and differentiation. This equation shows that if we talk about the development, then we have to discuss about the growth as well as the differentiation. Both are unitedly comes under the development. Now, it is under the control of both. Means development is control of both. Now, there are some intrinsic factors. right? Intrinsic factor include the intracellular elements like genetics, which determine the plant's potential for growth and development. Now, there are some intracellular that include the genetic. Intracellular factors involve the chemicals such as the plant growth regulators, which play a key role in coordination, growth, and development. Now, there will be some extrinsic factors that are crucial as they provide the external conditions necessary for optimum plant growth. Understanding these factors help you to create the better growing environment, right? So they will provide, it include the light, temperature, water, oxygen, nutrients, right? These factors are included as an extrinsic factors. Now we will talk about the plasticity. Plasticity is an important aspect of the plant growth and development that allows the plants to adapt to their environment. So, plasticity is an important aspect of plant growth and development that allows plant to adapt to their environment, right? How it is? So it can lead to the formation of different kinds of structure. Plasticity will forms a different kinds of a structure, right? Now, heterophily. Let us understand the plasticity with this particular example. Heterophily means two types of leaves. Developmental heterophily, the leaves of the juvenile plant, when the plant is very small, the type of leaf it possesses, it will be very different from the same plant when it gets matures. So mature plant is having a different form of leaf, whereas the same plant, when it was a juvenile stage, it is having a different form of leaf. So that is why it is called as developmental heterophily, right? So this type of like heterophilic phenomena we see in the some of the plants like the cotton, coriander, and larkspur. Environmental heterophily. Now, the shape of leaves produced in air is very different from those that is produced in the water. Like buttercup is the best example of this. If you see the figure, you can see here the first figure that is A1. You can see the juvenile leaf. What type of juvenile leaf it is there, and what type of leaf when it forms to the adult, when it becomes mature plant. You can see the leaf structure totally changed from its juvenile stage. This is for the development heterophily. The figure B, it is showing the two plant, the one that is growing on the land, that is the buttercup, Ranunculus, belongs to the family Ranunculaceae. And you can see when the plant is growing on the land, it is having what type of leaf? The B figure, the first one, terrestrial habitat. And the plant, when it is grown in the water, shallow water. So this shows the leaf very dissected type because the water current passes by it. So if the leaves are unitedly, like they are having the big lamina, then it can be dissected. The water current can turn that leaves. So they get modified their leaves in the form of very dissected form of leaf through which the water current can easily pass through it. So this is the very, very important phenomena, plasticity, that shows a type of development. Next, we will talk about the plant growth regulators, their characteristics. So plant growth regulators, they are very small, simple molecules, but they are having a very diverse chemical composition. We will see the different types of hormones and they are having very different chemical compositions. So they could be indole compounds, a very important indole 3-acetic acid, it abbreviated as IAA. Second, adenine derivative, N6-furfuryl aminopurine, commonly it is called as kinetine. Derivative of carotenoids, abscisic acid and terpenes, that is called as gibberellic acid. So carotenoids transforms and it forms the abscisic acid. Terpenes transform and it forms the gibberellic acid. And one of the very common gibberellic acid is GA3, because there are many types of gibberellic acid. It is extracted from the plant, and the one which is very common is the GA3. Gases, ethylene, C2H4. Also, they are described as the plant growth substances. Okay, these are called as plant growth substances, plant hormone, and also they are called as phytohormones. Why they are called as phytohormones? Because they are extracted from the plant. Characteristics. So based on their function in the living body, plant growth regulators are broadly divided into two categories. Let us see. First one is the growth promoters. The hormones that promotes the growth, that increases the growth, like cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. All these are shown as a plant growth promotion. So these are called as plant promoters, growth promoters. Now in this plant growth promoters, what are the different hormones that comes up? First one is the auxin. Second is gibberellins. Third, cytokinins. And fourth is the growth inhibitors that inhibit the growth. The two category we have already talked about initially. Like the plant growth regulators are broadly divided into two groups. The first group was the growth promoter and the second is the growth inhibitors, right? So in this involved in response to the wound or the stress, when the plant is facing some wound or some type of a stress, okay, biotic stress or abiotic origin type of stress. So they at this time, their activities is somewhat dormancy and abscission, right? These are the two types of growth inhibition that we see in the case of plant, dormancy and abscission. Now, in the growth inhibitors, the hormone that comes up are the abscisic acid and ethylene. Now, ethylene can fit in both the groups, right? But largely it is working as a growth inhibitors. Now we will talk about the discovery of the plant growth regulators. So first of all, we will talk about auxin, how the auxin was discovered. Charles Darwin, he was the son of Francis Darwin. Now they were doing some experiment taking the canary grass coleoptiles, okay? And this shows that when the light was passed on the coleoptile, all the coleoptiles start growing towards the light. So they are showing the phototropism phenomena. The coleoptile was a source of transmissible influence causing the bending. So this Francis Darwin and Charles Darwin, they observed that there only the tip, first the tip is moving. So what they do, they cut the tip. And what happens then? FW went isolated auxin from the tip of oat seedling coleoptile. And when the tip was cut, the coleoptile was not moving towards the light. So it means that the tip is having some chemicals in it. So FW went isolated a chemical from the tip of the coleoptile, and that is called as auxin. So here you can see. Now, the auxin was later, if you see the C1, the auxin was later put on this and the light was passed on it. So again, it is started moving towards the light. You can see in the D figure, right? So in this way, the auxin was discovered. Now, gibberellin, how the gibberellic acid was discovered. So Bacony fullest seedling disease in rice was caused by the fungus Gibberella fusariae. Now, this is a disease that occurs in the rice. What is the name of the disease? Bacony, commonly called as foolish seedlings. Now, this disease is caused due to the infection of a fungus that is called as Gibberella fusicori. So, E. Kurosawa in year 1926, he reported that some disease symptoms in rice seedling treated with the sterile filtrate of the fungus. Now, what they have done, they have taken out the fungus, they have taken out the extract, and when that extract was put on the seedlings of rice, the same disease symptoms appears on the rice plant. So, they have concluded that some active substances must be present in the fungus, right? So the active substances was later identified as gibberellic acid. They have extracted this active substances. How the cytokinin was discovered? Let us see that. So F. Skoog and his co-worker observed that tobacco stem internodal segment. Internodal segment means the portion between the two nodes, that is called as internodal segment, and that only proliferate the callus, that is undifferentiated cells. Now, if the nutrient medium, in addition to the oxen, was supplemented with the extract of the vascular tissue, the yeast extract coconut milk or DNA. These were the nutrient medium that were supplied to the extract. Now, Miller et al. In year 1955, he identified the crystallized cytokinesis promoting active substances that was named as kinetin. Kinetin was that the substances that he taken out as an extract, okay? And this is the substance that are responsible for the division of these cells. So next is the abscisic acid. So during mid 1960s, 3 independent research groups were working, and chemically they extracted 3 different, because they are working in a 3 different group. So one group has extracted inhibitor B, one group has extracted abscission 2, and another group has extracted dormin, right? 3 different inhibitors. Now, these 3 were later proven to be chemically identical and were named as abscisic acid, right? When they have tested all the 3 different types of chemicals, they concluded that they found that all the chemicals are similar. They are identical and they are named as abscisic acid. Next is the ethylene. And H. Cousins, 1910, confirmed that a volatile substance released from the ripened fruit, okay, oranges. He has taken oranges, ripened oranges, and from that, the volatile substances came out. And that volatile substances causes the ripening of banana. Orange were ripe, but when the bananas were kept with the oranges, they saw that the banana also got ripe due to the effect of these oranges, because some volatile substances are coming from the oranges and makes this banana as a ripened one. So this volatile substances later identified as a gaseous plant growth regulator, that is called as ethylene. Now, physiological effect of plant growth regulators. First one, we will talk about the auxin. The term auxin. What is the meaning of auxin? To grow. This was first isolated from the human urine, right? So later, when it was isolated and when it was tested, it came that it is an indole-based compound, and the chemical name is indole 3 acetic acid, abbreviated as IAA. So this is the natural auxin, but there were some synthetic one also. So they are primarily produced in the growing apices. Now the point comes here, at which place in the plant these plant growth regulators were, like auxin, first one, where it is produced. So it is produced at the apex of the plant, both at the apices of the stem as well at the root, right? And from there they can migrate to any places where there is requirement of the auxin. Natural auxin include indole acetic acid, right? And indole butyric acid. These are the two natural examples of the auxin. The synthetic one, naphthalene acetic acid and 2,4-4-dichlorophenolic acetic acid. This is the two examples of the synthetic auxin. So all these auxin are used extensively in the agriculture and horticulture practices, right? These are extensively used in the agriculture and horticultural practices. So rooting. These hormones are very important for rooting. When we take out the stem cutting, and if you remember any example that is used for the stem cutting, that is the rose. In rose, the stem cutting that is used for the grafting method, right? So when you take out the stem cutting of the rose plant and dip in the auxin, you see that from that particular part which was dipped in the auxin, root comes very fast. Okay, so that could be used for the plant propagation. Flowering, auxin also promote flowering, and the example is the pineapple. Abscission control, they do not allow the plants, okay, to drop their leaves in the early stages. The fruits, they do not allow the roots to drop in the early stages. Fruit and leaf, they get intact to the stem because of the control of abscission. Promote abscission, but sometimes they promote abscission also, like shedding of the older mature leaves and fruits, right? They do not allow the young fruits and leaves to get detached from the plant, but yes, they allow the mature fruits and leaves to get detached. Apical dominance. Now, in higher plants, the growing apical bud inhibits the growth of lateral buds, right? So that is the examples of the apical dominance. Decapacitation. You remove the shoot tip. Okay, what happens? The source of auxin produced at the apical bud get removed. Now, the branches from the axils of the leaves comes out, right? So this reduction in auxin concentration at the lateral buds removes the inhibitory effect, allowing them to grow. The lateral buds will grow and forms the bushy appearance of the plant. So here in the figure, you can see the apical bud. When this apical bud got removed, okay, the B figure now you see, the branches are coming there from the axils of the leaf, new lateral buds are coming out. Now let us see the application. The principle is widely applied in the tea plantation. You must have seen that the women are plucking the top particular leaves of from the shoot, right? So this gives rise the lateral buds to grow more, and it forms the hedge, right? Parthenocarpy. Now this is a very, very important. Auxin-induced parthenocarpy means the fruit takes the development without fertilization. The fruits are get developed, but without any fertilization process. And the examples are tomatoes. Herbicides. They are widely used as herbicides. 2,4-D is very common herbicides. Means it can kill the herbs, but it will not harm any plant, your crop plants. So 2,4-D is specifically used to kill the dicotyledonous weed. When you are growing your monocot plants like rice and wheat, the weeds that are the dicotyledonous plant coming out from the soil, when you are growing your monocotyledonous crop plants, rice and wheat, so at that time when you spray the 2,4-D, your dicotyledonous will die. But there will be no effect on your monocotyledonous plant, that is rice and wheat. Now, so gardeners are using this 2,4-D very frequently to prepare their weed-free lawns, right? It also promotes the cell differentiation. So auxin control the xylem differentiation, right? Cell get differentiated. Earlier, due to the meristematic activity, they divide and redivide, but there are different types of cells, right? So they get differentiated. They get differentiated, and this is due to the auxin. Cell division. Auxin helps in cell division as well. Now we will talk about gibberellins. So gibberellin is a type of plant growth regulator that promote various aspects of the plant growth, right? Like if you see the diversity of this hormone, there are 100 known gibberellins, right? Found in the organism ranging from fungi to the higher plants. So if we talk about the 100 of gibberellins, here are some examples mentioned. GA1, GA2 and GA3, and out of which this GA3 is very, very common, right? So gibberellic GA3, this is the more first discovered, and this is the very extensively studied gibberellin, because every plant is having this type of gibberellin, right? Acidic nature, as you all know, that it is, the name indicate gibberellic acid, so it is having some acidic effect. Now, wide range of responses we see in the from the gibberellin. The first one is the stem elongation. Now, they are used to increase the length of the grapes stalk, right? Whenever there is a cropping of the grapes, they increases the length of the grape stalk, and that gives you the more yield as well. Spraying sugarcane with the gibberellin significantly increase the length of the stem, and that could also increase the yield of the sugarcane. So here is one data that it increases the 20 tons per acre. Suppose in a year, you are having 1 acre of land and you are getting 50 tons per acre, right? Next year, you will get how much? It is added 20 tons per acre, means 70 acre you will get. 70 tons at that year you will get when you will apply this gibberellin hormone. Fruit development. Gibberellin causes fruits like apples to elongate and improve the shape, that increases the commercial value in the market. Delay senescence. So the delay the aging process of the senescence is the aging process of the plant. So it delay that process, allowing fruits to remain on the tree for longer period. So thereby extending the market period. In this way, the market period get extended. Malting process, very, very important. Gibberellic acid is used in the brewing industries as well. This accelerate the malting process in it. So that is why it is increases. It hastens the maturity in the conifers. Spraying juvenile conifers, because the conifers takes a lot of time to get developed. So if the conifers were sprayed with the gibberellic acid, it speed the maturity period. Promote bolting. So it promote bolting, rapid internode elongation just before flowering in the plants. Examples, if you see, beet, cabbage, and many other roset growth habit. So here you can see the untreated plant, very short. It's showing the roset habit. But when you spray it with the gibberellin, you can say the internode region of the plant get elongated. And the roset habit is not there. So this is called bolting. So it promotes the cytokinesis, means the cell division. Discovered as kinetin, it modifies from adenine a purine. Adenine is an example of a purine nitrogenous basis, right? So first time it was discovered as a kinetin, and it is also derived from the herring sperm DNA. When the herring sperm DNA was autoclaved, this hormone was derived. So kinetin does not occur naturally in plants. It is occur in animals. Now in the animals, this cytokinin was obtained as a zeatin, basically from the corn kernels and the coconut. The natural cytokinin that produce areas of rapid cell division. Cytokinin is required in the plant for the cell division. Now, what are the examples? Root apices, developing shoot, but wherever there is a requirement of the division of the cells, like meristematic cells are very actively dividing. So there is a requirement of this hormone, right? So root apex, development of the shoot buds and young fruits. So it helps to produce new leaves. New leaves, they require the division of cells. Chloroplast in leaves, lateral growth of shoot, and adventitious root formation. All these regions are having this particular hormones. So it also helps to overcome the apical dominance, promote nutrient mobilization. So when there is a requirement of the mobilization of the nutrients from one region to another, there is a requirement of this hormone, right? It also delay leaf senescence. Now, next hormone is the ethylene. This is a very simple gaseous plant growth regulator. It influences horizontal growth of seedlings. Okay. Seedlings shows the horizontal growth because of this particular hormone. Swelling of the auxins, apical hook formation in the dicot seedlings, right? So it also promotes the senescence in the abscission of plant growth, especially leaves and flowers. So it promotes senescence. The growth regulators delay senescence, but this one is promoting the senescence. So in this respect, this is the plant growth inhibitor, right? Highly effective in the fruit ripening because it affects the fruit. Means if the raw fruit are kept with the ripened one will release this gas hormone and it makes the raw one also get as a ripe one. So it enhances the rate of respiration. That is called as respiratory climactic. It breaks seed and bud dormancy. Now, ethylene also initiates the germination in peanut seeds. In some cases, you see that it is initiating the germination process like in the peanut seed and sprouts of the potato tubers. You must have seen the potato tubers, they are having eyes and the sprouts are coming out of those eyes. So this is just because of the ethylene hormone. It also promote the rapid internode and petiole elongation, right? In deep water rice plant, specifically for this plant only. It helps leaves upper part of the shoots to remain above water. Always the upper part of the plant, that is, remain above the water. Promote root growth and root hair formation. used to initiate flowering in the pineapple and mango. See, this is helping in the flowering for pineapple and mango, right? Ethaphone. Now this is chemically available in the market as well. Ethaphone is widely used in commercial source for the ethylene. So whenever you required this ethylene hormone, you can get it from the market as in the form of ethaphone. Once absorbed, it transported throughout the plant, right? So within the plant, ethaphone slowly release ethylene and it makes uh, the fruits to get ripen. So ethylene releases leads to several effects. Like first one is the fruit ripening, example in the tomatoes and apples, formation of abscission, flowers and fruits leading to thinning in cotton, cherry and walnut. It also promote development female flowers in the case of cucumber, right? It can increase their yield. Next, plant growth regulator is the abscission. So regulate abscission and dormancy. It promotes the dormancy, like we have heard about the dormancy of seeds and abscission layer formation in the leaves. So this is because of this abscisic acid formation. Act as a general plant growth inhibitor. It is working as a plant growth inhibitor. Inhibit seed germination. Do not allow the seeds to get germinated because of its effect as a dormant, means dormancy of the seeds. Stimulate the closure of stomata in the epidermis. Do not allow the epidermis cells that are modified into the stomata. So it do not allow the stomata to get open, right? So in their stress condition, it allows the stomata to get closed. Water stress condition. So increase the tolerance against the various stress like the water stress. So that is why it is called as stress hormone. It plays a very important role in the seed development, maturation and dormancy of the seed. It keeps the seed dormant stage, right? So it is working as an antagonist to the gibberellic acid. Let us see some more effect of abscisic acid. So plant growth regulator are crucial for all plants possesses. Like every phase of plant growth, there is a process of differentiation, development, right? And there is a requirement of at least one plant growth regulator, right? All the processes require plant growth. Now, there are some plant growth regulator interactions. The role of plant growth regulator that can be complementary or antagonistic, right? One is working just opposite of the other plant hormone. So they can work together or oppose each other, right? So individualistic or synergistic, they can act alone. Some hormone act alone, but some hormone join together and then work to one particular function. So they are showing the combined effect of both the hormones. Multiple plant growth regulator affect key events. Let us see that. Seed bud dormancy, abscission, senescence, and the apical dominance. Many of the plant hormone, okay, do this particular function. So they are working as a synergistic, showing the synergistic effect. So they are influenced by the interaction of more than one plant growth regulator. So integrated control of plant development. Plant growth and development is governed by the combination of first one, plant growth regulators, genomic control, and some extrinsic factor. Not all the intrinsic factors are working there. Like intrinsic factor are plant growth regulator that is formed inside the plant only. Genomic control comes from the plant only. But there are some extrinsic factors as well, like temperature and light. They often control the plant growth, development, and influence their plant growth activity. So examples of plant growth regulator that shows the extrinsic factor: vernalization, flowering, dormancy, seed germination, and various plant growth movements. Okay, let us see the summary part of today's session. Plasticity. So plant follow the different pathways in response to the environment. That is the one example we have seen. That is the heterophyly. Okay. Sum of processes, development we have talked about, growth and differentiation unitedly comes under the development. Now both internal, that is the genetic and plant growth regulators, and external factors like light, temperature, water, and nutrients unitedly working together to bring about the development. So plant growth regulators, we have discussed about the different phytohormones, simple, naturally occurring, and they regulate the different physiological activities. The 5 major groups we have discussed today: auxin, gibberellins, cytokinin, abscisic acid, and ethylene. So auxin, gibberellin, and cytokinin, these are the plant growth promoters. So they brings about cell division, elongation, and differentiation, whereas some are the inhibitors like abscisic acid and ethylene. They are primarily involved in the dormancy of seed, senescence, aging in the plant, abscission, layer formation, and some stress responses. So ethylene, this can be as a promoter as well as an inhibitor. We have talked about the synergistic and antagonistic effect. Some hormones, 2 or more hormones, unite together and bring one particular effect, whereas some hormones just bring the opposite effect, that is the antagonistic effect. Now let us see the NCERT based question. Which of the following statement about the auxin is incorrect? Let us see the options. A, they promote cell elongation. They promote apical dominance. C, they promote root initiation. And D, they promote early flowering. Let us see the answer. D, they promote early flowering. This is the correct one. Now let us see the explanation part. So they promote the cell elongation in stem, right? And they maintain the apical dominance by suppressing the lateral bud growth. Only the apical buds are growing. So they promote root initiation, especially in the stem cutting. However, auxin do not promote any early flowering. So flowering is regulated by the gibberellins. Next question. The senescence in plant is promoted by. Options are A, auxin, B, gibberellins, C, cytokinins, and D, ethylene. Let us see the answer. D, ethylene is the correct answer. Okay. So senescence refers to the aging process in plants leading to the death of tissue or organs. And ethylene, this is the aging hormone or the ripening hormone. It promotes what? Ethylene promotes ripening of fruits, abscission layer, formation in the case of leaves and fruits, senescence of flowers, aging of tissues. Basically, it brings aging in the plants, right? Now, let us see some keynotes. Auxin were first discovered in the oat coleoptile. We have seen this in the discovery part of the auxin. Gibberellins were first discovered in the Japan from the rice disease, foolish seedling. Ethylene is only gaseous plant growth regulator known. Abscisic acid is also known as stress hormone. Coconut milk is rich in cytokinins, right? Now let us see some interesting questions. What happens if you apply auxin to one side of a plant stem? Why do the pineapples flower uniformly when ethylene is applied? Third, how does a plant know when to shed its leaves? And fourth, Why do seedless fruits often require gibberellin treatment? So with this, we end up our session. Thank you very much for attending today's session.